Welcome back, students. Today, we are starting something very important in biotechnology, the recombinant DNA technology. Now, this chapter is foundational. If you understand this properly, everything else in biotechnology becomes easy. In this video, we are going to understand what recombinant DNA technology is, what gene cloning means, what a host is, what a vector is, and the different types of hosts and vectors used in biotechnology. So let's begin. So first, what exactly is recombinant DNA technology? Recombinant DNA technology refers to the joining of two different DNA molecules with the aim to isolate, propagate, characterize, and manipulate genes for various applications. We are joining two different DNA molecules. This is the heart of this technology, and this entire process is also called gene cloning. Gene cloning simply means making multiple copies of a desired gene, not cloning a human, not cloning an organism, cloning a gene. Now let's understand the process step by step. We begin with two things, a vector and a piece of foreign DNA. From the foreign DNA, we identify the target gene, also called the insert. Both the vector and the foreign DNA undergo restriction digestion. That means they are cut using restriction enzymes. After cutting, we join the target gene with the vector using a process called ligation. Now the vector contains the target gene. This is called recombinant DNA. Next, this recombinant DNA is introduced into a host cell. This step is called transformation. Inside the host cell, the recombinant DNA exists separately from chromosomal DNA. Once inside the host, the recombinant DNA undergoes replication. As the host cell divides, the recombinant DNA is multiplied. So eventually, we get many copies. This entire process is called gene cloning. Now, here is something very important. Recombinant DNA technology is also called a two key component system because without these two, it cannot work. The two components are the host and the vector. So, what is a host? A host is an organism into which recombinant DNA is introduced. The host supplies all the required materials for smooth replication of vector. Now pay attention to this. The host provides enzymes, proteins, and genetic machinery required for replication and expression of the recombinant DNA. Without the host, the recombinant DNA cannot function. Hosts can be divided into two major categories, prokaryotic and eukaryotic. The most extensively used prokaryotic host is Escherichia coli. This is a rod-shaped gram-negative bacterium. It is commonly found in the lower intestine of warm-blooded organisms. The K12 strain of E. coli is widely used in gene cloning. Now here's why it's popular. It reproduces and grows rapidly. It has a doubling time of approximately 20 minutes. That means every 20 minutes, the population doubles. Other prokaryotic hosts have also been developed. For example, Bacillus subtilis. It is used when the aim is secretion of a protein encoded by a cloned gene. Among eukaryotic hosts, yeast is commonly used. It serves as an important host in gene cloning. Now let us understand the second component, the vector. A vector is a DNA molecule which can replicate itself inside a host cell. It is used to carry the target gene into the host organism. Think of it as the carrier. For a DNA molecule to act as a vector, it must fulfill certain essential features. Number one, it should have an origin of replication so that it can replicate inside the host. Number two, it should have at least one selectable marker, such as an antibiotic resistance gene. Number three, it should have unique restriction sites for insertion of foreign DNA. Number four, the vector should be small in size. These are essential. The vectors can be divided into prokaryotic vectors and eukaryotic vectors. Prokaryotic vectors include plasmids and bacteriophages, and the eukaryotic vectors include yeast, plant, and animal vectors. In the upcoming videos, we'll go deeper into the different vector systems. We will study the plasmid vectors in detail, bacteriophage vectors, and how specific vectors like PBR322 and PUC19 are structured. Also understand how cloning actually happens at the molecular level. So this video gives you the foundation, and the next videos will build the structure. I'll see you in the next lesson. Thank you.